The overall goal of the following experiment is to achieve site-specific transfection in adherent cells. This is achieved by culturing a monolayer of cells on a microelectrode array. As a second step, the application of an electric pulse to select electrodes leads to electroporation of the targeted cells adhered on top of the electrodes. This leads to the creation of pores in the cell membrane that allow the entry of exogenous molecules into the cell. The targeted cells can then be screened to detect the resultant phenotype using an appropriate assay.
The microscale transfection technique we describe here has several key advantages over conventional transfection techniques such as bulk electroporation or chemical transfection. A, it allows us to achieve exquisite spatial control over the transfection process itself and helps us to transfect single cells or cluster of cells with high efficiency even in difficult to transfect cell lines. B, it is an approach that scales up readily and helps us to realize high throughput molecular assays.
One of the key advantages of using a microelectrode array for site-specific transfection is that it enables simultaneous functional assessment of targeted cells, such as quantitative electrophysiology and morphological changes.
To begin the procedure, sterilize the MEA in an autoclave. Then, transfer the autoclaved MEA to a laminar flow hood and place it in a standard sterile polystyrene petri dish. Prior to seeding the cells on the MEA, sterilize the laminar flow hood by turning on the UV light for 20 minutes. If the MEA is sensitive to UV light, cover the petri dish containing the MEA with sterile aluminum foil while the flow hood is